Hello everyone, I am Professor Kazi Ahsan Habib from Bangladesh. First, I'd like to give my thanks. Maybe my voice is loud, I think you can hear. Anyway, so I would like to thank ANEMONE Global to invite me here. So today I will present actually my DNA barcoding and eDNA metabarcoding work in Bangladesh. Firstly, I will introduce shortly with the maritime area of Bangladesh. Then I will show how collaboration and connectivity connectivity between my lab and other organization led me to dive into this type of sophisticated work, especially DNA metabarcoding, also DNA barcoding, being a developing country. So sometimes it is tough to arrange all different types of technologies in lab, but how collaboration made me able to start this work. So if I see, this is Bangladesh maritime area. So we have got 118,000 square kilometer almost in the part of the Bay of Bengal. So Bangladesh is located in the top of the Bay of Bengal between India and Myanmar. So, and our coastline almost 710 kilometer. So this is the Bangladesh maritime area. So if you see the position of Bangladesh, the two large river system, one is Ganges river system, another is the Brahmaputra river system. These both river system flow into the Bay of, fall into the Bay of Bengal, flowing over the country. So Bangladesh, in fact, is a deltic country, deltic island. So yes, and so this is very one of the most fertile land in the world. So this is this big two river. So this is then if we see our coast, if our estuarine ecological coastal and estuarine ecological region, we can divide it into three parts. If you see the easternmost parts of here, that is the Ganges, westernmost part, you see the Ganges tidal floodplain, that is basically mangrove region. And then if you see the middle part, this is the Meghna deltic island. Actually, this is the major estuary in Bangladesh. And if you see the right side here, the eastern part, that is the Chattogram-Cox's coastal plain, that is basically made of the sandy beach and sandy area. And the other part, the middle area, is very, the high siltation occurred in the middle side, and the western side, that is the water is very turbid with muddy. So this, if you see, I will show you some important areas in our maritime area, some important zones. You see, this is the marine protected area that was declared by Bangladesh government, total 5. So these are very important for biodiversity. Then this is the swatch of no ground, middle ground, and these 4 fishing ground in Bangladesh. And then if you see, this is the breeding area of our national fish, Hilsa. So lots of people are involved with their livelihood to catch this fish. This is a migratory fish actually. It moves to the river water for their breeding purpose. So then if you see, this is the Sundarbans area. You know, this is the largest single tract mangrove forest in the world. So, and then this is another swatch of no ground. This is a submerged canyon, almost from 900 to 1200 meter depth in this area. So, and this is another very important area for biodiversity. And this side, there is the Moheshkhali, Sonadia Kutub, there are some islands are there. This is another ecologically critical area for Bangladesh and important for biodiversity. Now, I will show actually DNA, my lab, ABR lab, Aquatic Bioresource Research Lab. We are actually, in my lab, we assessed the marine biodiversity of different ecosystem of Bangladesh. So what are we doing? We are preparing DNA barcoding reference data. That is, we assessed and we are assessing marine biodiversity using DNA barcodes. And also at the same time, we are making the DNA barcodes reference database. And then in identification of unreported and new species during when we worked with DNA barcode. And then environmental DNA metabarcoding using mitochondrial DNA genome. This is relatively new work in my lab. So that we are working now. So this is first, if I see the Sundarbans ecosystem, you see this is the Sundarbans, world's largest single-tract mangrove forest. So it is also UNESCO World Heritage Site. And we know that this Sundarbans is very nursery and breeding ground of fish and shellfish, specifically crustaceans, shrimp, crab, and many fishes. So it is almost 10,000 square kilometer, and 60% belongs to the Bangladesh and 40% in the West Bengal of India. So this is so we call people call it the Amazon of the East. It is also a Ramsar site, a very important Ramsar site. And during my DNA barcode work, when I started, actually after finishing my PhD from Korea, when I came back to Bangladesh, I started this type of work. Then we found several types of, new species. This is the one new species to science. We identified from Sundarbans. We also confirmed confirmed the species identification of mud crab in Sundarbans. You know, four species are found in the world. So we were confused which one is that. By DNA barcode, I confirmed that, and now the breeding and other works are going on this mud crab. Our main mud crab is the Scylla olivacea. Previously, it was assumed Scylla serrata. Then we published DNA barcode of the fish and shellfish of Sundarbans in PLOS One. We found in this paper we accumulated 113 fish species and other shellfish species like shrimp, crab, and some mollusk. So that is, I published one book on Sundarbans biodiversity. That is basically is a taxonomic book. So we also gave the real picture that we captured in the lab and other different types of fish, arthropods, mollusks, amphibians accumulated with the DNA barcode accession number of gene bank that was made by my lab. So that is, we published with the help of South Korea, YOSU project, and actually it was also collaboration with my supervisor, Professor Yeon-Ho Lee. So we collaborated in this work. Then we published this, and then now I am doing DNA metabarcoding. Oh no! Later on, in that collaborative work, we also made DNA barcodes of zooplankton, eggs, and larvae. So we collected these by using zooplankton net. We collected larvae and others egg, and this is the sampling spots. You know the sampling spot. So then you see the clear difference. It was my first attempt at DNA barcoding of larvae and other zooplankton. So in January, basically in the winter, that area was dominated by the arthropods and also some echinoderms. And in the next year, when the summer started, it was dominated by the fish. So basically, the fish and other organisms. So that was my first work. Then, anyway, so then maybe a couple of years back, I met Professor Kazita in a online meeting. So he's very kind. Then I told him I want to join in your team. Then I joined with Kajita Sensei's team. That is still, I am working with him in his collaborating team. So then I started eDNA, started to assess fish diversity with eDNA metabarcoding. So this is, you know, my first sampling with the kit sent by Kajita Sensei, the Sterivex. So we used the 5 Sterivex only, that we got 55 species from my first attempt in Sundarbans. Then later on, I also worked with the UNESCO environmental DNA expedition in the first phase. So I joined, I lead that work in Sundarbans. So UNESCO, you know, the UNESCO has taken the eDNA expeditions in around the world, basically in the World Marine Heritage Sites. So Sundarbans is one of them, marine heritage. So I worked with the UNESCO. See, so from UNESCO, if you see, anyway, this is the sampling spots, 5 spots from the middle of the wildlife sanctuary. I collected from the 5 spots, so eDNA. Then we found almost 113 fishes, 4 mammals, shark and rays 6, and others. So they published it in the OBs and in some magazine. So I am working to write the paper on it. And this is the results of order-wise distribution, then family-wise distribution. And this is some news on the Sundarbans work in eDNA expedition in the UNESCO website and in my local newspaper. So future plan of study. So I will continue the work already Kajita Sensei told in JSPS Core to Core program in the mangrove region. So this year I took a PhD student to work in the long run work, to maintain the long term work. And I also expressed to work with the UNESCO phase 2 eDNA expedition. So I showed the then this is the Cox's Bazar area. So from this Cox's Bazar area, with the Michio Sensei, I also take some samples from the Cox's Bazar region to work here. So this is the area. So you know, the Cox's Bazar is the longest sea beach in the world, a single track longest sea beach, very slopey, 120 kilometers almost. So I collected from 3 stations, and this is the results. So these are the results, and this is the top 10 breeds of fish species. And this is the St. Martin's Island. We have very small but very important island, coral island in Bangladesh. So I also worked in the DNA barcoding and metabarcoding work. This is the some underwater picture of St. Martin's Island taken by my lab's member. So this is, we reported first 12 coral species this island, and this is the publication of DNA barcoding of reef-associated fishes from St. Martin's Island. So we barcoded 84 reef-associated fishes here. And this is some picture of the St. Martin's Island picture, and we discovered a new species from St. Martin Island in our DNA barcoding work. So this is the first attempts in St. Martin Islands. The result is not very good, but later on we again collected samples. With the ANEMONE Global, we collected the samples, water samples, eDNA, and sent to the Japan. Now it is under work. So this is one book we published, taxonomy books on the reef fishes of St. Martin's Island with DNA barcodes, and that is here. And then this is the underwater album that we published with the help of IOCO spec, and I sincerely thanks to the Uzi. Actually, he arranged the money for publication fees. He gave me the publication fees charge of this book. So then, and finally, actually, I made a online database of my marine fishes in Bangladesh. So I accumulated, I reviewed from 1971 to until now. That is my independence, year of the independence of Bangladesh. Until now, all the species I reviewed and listed and put it into here, and also the picture, maybe some secondary pictures are there, so that people can easily get what we have the marine biodiversity. Because now it needs different books to see different books and different reports. So I accumulated here. So this website you can visit. So I am almost finished. This is the different pages of the website, different taxa. If you click here and go inside and see page to page and some basic data, the Bengali name, common name, scientific name, classification, the characteristics, and some fish-based related some different website. So that is and that is the anemone. In my future plan to continue work with ANEMONE Global, especially with the MPAs. So to monitor the MPAs, assessing the biodiversity MPAs and changing the diversity, I want to continue with ANEMONE Global. Here is the swatch of no ground. I want to work with anemone. This is some swatch of no ground. This is some pictures of whale and dolphin, and this is Nijum Deep, another area, the single sex, that is an MPA, and this is also Naf River, another MPA. So I want to actually continue my work with the ANEMONE Global. So thank you very much, and I acknowledge this organization for my work and collaboration. Thank you very much.
Okay, thank you very much, Habib-san. Yes, we can take very quick questions.
Okay.
Definitely so.
Are the sequences being registered also in a public domain such as GenBank, and are the samples, I mean the actual paratypes, is also being registered in a museum, so related to the actual sequences? Thank you.
Yes, actually what we discovered or detect the DNA barcodes, we submitted in the GenBank and BOLD database system, and this year I got a fund for museum. So I am making a museum in my lab, not in my lab, in my school. So I have all the samples now under the preservation. So I will arrange this in the museum. So this year government gave me the fund. So I can make my own museum. Actually, Bangladesh has no historical natural history museum until now. Some sporadic, but I have planned to collaborate with the government, also my school, and then make a very good central museum type. That is my aim in the coming couple of years. Thank you.
Thank you.